Hey everybody. I am gonna go through the lecture today. Sorry it's taking me so long to get this posted. I appreciate your flexibility while we're going through this. So I'm gonna share my PowerPoint for you. I'm gonna go through the material. If you have questions, then go ahead and write those down and then come see me on Wednesday. And then also I have posted inside the assignment, the in-class paper. You're going to have to probably print those off and then you can submit them as you normally do if we were in class. Again, this is cell transport. It is probably the easiest one out of all of the topics that we're covering for this test. Um, and I have included um, the quizzes and stuff already for the test in 2 weeks. You guys can go in and start practicing on those, and then I will have the test review up probably by the end of the week. All right, so let's just get into it. I'm going to share my screen with my PowerPoint. Hopefully you can see that okay. Let me actually do the slideshow. Okay, so, oh, that's probably not the best one. Let's just do this. Let's just play it from current slide. Still doing it. Hold on. Sorry, guys. It's just being dumb. Okay. Um, Try one more. I'll just do it this way. Okay, so this is the like I said, cell transport. So we're looking at a review of what we talked about last week. So we're looking at cell membranes and fluid mosaic pieces of the lipids and proteins. So we have our glycoproteins. Remember, you guys were supposed to label those last week. Basically, we're just talking about how everything is able to move in because that has to do with transport. And so looking at this, we have a couple of things that we've labeled as of last time. We have our integral proteins. There are two types of that. There's channel proteins, and then we'll get to the other type. And then these are our glycolipids. They're a signaling piece so that we're able to tell or be able to signal out for things that the cell might need. Your cholesterol that keeps the mosaic fluid piece going along with the cell membrane to let things in and out rather quickly. And then, of course, the structural piece of the phospholipid bilayer in general. I do this there. How's that? Hopefully that's a little bit better. So looking at diffusion, this is the first one. So we're looking at two types. We're looking at the passive and non-passive. So active and passive. Sorry, it's been a long day. So when we're looking at diffusion, diffusion requires a gradient. So if you're looking at these two guys right here, we have particles. Diffusion is also particles and it's not water. So anything that is particles, in this case, the orange is particles of water. So it requires a gradient, which means it goes from high concentration to lower concentration. So if we're thinking about how that will work, if I'm at the top of a slide and I'm going down, it goes from the high concentration to the low concentration. The slide does not require me to have energy to get back down the slide. In other words, I need a ton on this side and for it to go. So this is the gradient we're looking at. So if I'm trying to decide which direction this is going to go, which way do you think it will go? Hopefully you say that it's going to go from the higher concentration down the slide to the lower concentration. And then that way the particles are moving where they need to go. It goes in that direction. So when we're looking at diffusion, we're moving down the concentration gradient that is depicted by this. So we're going down the slide, going from high concentration up here to a lower concentration. No energy is needed. This is passive. So if we're going like that, little guy, and all of these suddenly, once we get that to that point where everything is moved to where it is hits equalness, it's called equilibrium. And then we have a net zero movement, which means that it's still moving, but it doesn't, the movement from one end to the other is basically at zero because what's coming to this side and what's going to that side is equal at the same time. Transportation facilitated diffusion is actually passive as well, but it needs help. It usually will have movement through molecules like the protein channels that we talked about last week. This still doesn't need any type of energy, but it is using those transport proteins that we looked at in the cell membrane in order to move from the higher concentration to the lower concentration. Those transport membrane can be a protein channel, a carrier protein. Channel proteins are basically just allowing things to move back and forth from the extracellular space to the intercellular space. This is outside of the cell versus inside of the cell. And those are usually hydrophilic substances that are moving across the membrane. So you have two different types. You have the channel protein, which is the integral protein, which means it's embedded in carrier proteins are also embedded, but they have kind of a little bit of a trick to them. They actually check the size. This is the one that I talked about last time that's a little bit more specialized. But this one will just kind of let whatever needs to go through go. And we're still moving from higher concentration to lower concentration of particles, which is the green dots. And some of these are based on size. A channel protein has the hydrophilic channel that allows those molecules or ions that can be used that can go through the tunnel. So again, this one is a little bit less tricky because it as long as it can fit through the tunnel, as long as the cell needs it, it can move through that channel. It's not as specific as say the carrier protein where they are needing to change shape to move things across the membrane. So one of these things would be maybe glucose. And they have carrier proteins binding to molecules themselves to make sure that they are the correct thing, and then they pull them through. But it is still high to low concentration, higher concentration to lower concentration. And that means that there is still no energy needed for this process to take place. So if we're looking at facilitated, it still works down the concentration gradient from the high to the low, and it's still going to be going through the membrane, except for now it's getting help through the carrier or the channel protein. So it's going to move from the high concentration through that channel protein one at a time until they're based and balanced out, till this gets to the point where they are in equilibrium. All right, osmosis is a different thing. Osmosis is the transfer of water from a higher concentration of water to a lower concentration of water. So if you look at this example, you have a ton of, let's say this is glucose, a ton of glucose here, and very little here. There's less water in this area than there is this area. So the water is going to basically move through the semi-permeable membrane until it is balanced out, where the water ratio matches the diluted, the version of the particles that are there. Specifically, in this case, we said it was going to be glucose. Sorry, I got a little tickle. All right, so if osmosis of diffusion of free water across the simmer, so these are osmosis pieces. So if we are going from a high concentration of water to a lower concentration of water, and now these particles are water themselves, water is going to, I am going to rephrase that, sorry. So the water is blue and the orange is the particles. They are not moving. This is a semi-permeable, selectively permeable membrane, which means that the orange can't fit. So we're going to have water. If you notice that there's tons of water on this side versus very little water here. So it's not moving from this direction to this direction. It's actually moving the opposite way. So it's going from a high concentration of water to a low concentration of water. The reason for that is that this permeable membrane doesn't allow, it's too big for it to go through and balance out. So the next best thing is to have that water move through the other direction in order to balance out the water and to give those particles a little bit more of an equilibrium. So the water, because it can move through that membrane, is now moving across to kind of balance out that side. So now we have almost the same amount of water on both sides. And again, once it hits the point where it is equal, it is now in equilibrium. There are two types, or two different levels of tonicity. I know that there is three, but isotonic is basically a balanced out version. So that would be your equilibrium. And then when we're measuring something as far as tonicity, there are two things to do, hypotonic and hypertonic. So if we're looking at hypertonic, hypertonic means that there is more particles in the outside of the cell. Let's say that this is a blood cell and this is just water. With some salt in it. So let's say that this here is 10% salt and inside of the cell is only 1% salt. So there is more water potential or more water here than there is out there because those large amounts of salt are basically clogging up the water on the outside. So to balance it out, water is going to move from the cell out to the water, causing the cell itself to shrink. So if we're looking at a blood cell, it's going to shrink up. If we're looking at a plant cell, it'll still maintain some shape, except for it will become turgid, which means, or not turgid, plasmolysis, because what happens in plasmolysis is it will shrink the cell membrane area, and then the outside cell wall will become like very weak, and it will become like very, very flimsy. Isotonic, again, water is equal on both sides. So this would be like a 1% water. This is 1%, or I'm sorry, 1% solute, 1% solute here. They're equal. It's a net zero movement. On the flip side of the hypertonic, where the cell is going to shrink, we have what we call hypotonic. And hypotonic is where there's more water on the outside of the cell versus water inside of the cell that matches the solute. So let's say that this is 1% solute or salt inside of that cell. And then out here, let's say it's 0. Let's say it's just tap water, it's almost nothing is in it. And so the water out here, because it wants to balance these two areas out, is going to move inside of the cell. If it's a plant cell, it becomes turgid, which means it gets really tight in there. It becomes crispy. If it's, say, like celery is an example of that. If I put celery into a distilled water bowl, then the celery itself is going to become crunchy because the water's moving in and trying to balance out. But if I put celery into salt water, the water from the celery, because there's less solute inside of the celery than the salt water, is going to go back down to this direction where the water is going to move outside of the celery, and this is going to become flimsy, and all of the water will go into the beaker with all of the salt. So for this one on hypotonic, it is moving into the cell, which for a plant cell is not a bad thing because they can monitor that. But for a animal cell, it becomes an issue because there is nothing to protect that cell. So what happens is the water goes in, it's trying to balance out that salt potential, and then it explodes. So it lyses. And so when we're looking at things like blood cells, that's a big issue because we do have, when you're looking at the combination of what's going in, say, to the cells themselves, think about this. If I am getting an IV because I'm dehydrated, they're not putting in distilled water into your vein. They are putting in a combination of the salt potential that you have in there. So blood is usually 0.9% sodium chloride. And so a saline IV is going to be around that too. So putting just straight water into your blood vessels will cause your blood to actually become hypotonic or lyse, which means they're breaking apart, and then that's a problem. All right, so if we're looking at blood cells itself, here's our hypertonic. They're so hyper that they jump up and down and lose weight. This is where the water is going to be moving out because the outside area of the beaker, let's say this is a beaker, has a higher concentration of sodium. And so that water from the cells themselves is moving out. So let's say this is 0.9, and then out here is 10% sodium. So the water wants to balance out, so it moves out of the cell. Isotonic, again, what comes in goes out. It's very equal. the cells are staying intact. And then when we're looking at hypotonic, the combination of it being at 0.9 versus 0%, let's say this is 0% distilled water that the blood cells are being put into, becomes the water's moving in because it wants to balance out, but there's nothing protected. So then it becomes lysed. So they explode or blow up. And of course, we have issues if they're human cells and you're trying to give water into a vein. So we're going to watch a quick video. It goes from isotonic down here. It'll tell you it's isotonic versus hypotonic. Right now we're looking at isotonic, which is very well maintained, and we're just watch it for a second. So we're going to watch, there's our isotonic cells. And then we are now looking, everything looks pretty normal. Nothing has changed, but now we're putting them in hypertonic, which means the outside solution has more salt. So watch the cells as they start to shrink because the water from the cell is trying to balance out, and now they become like little raisins. So they're becoming now we're going back to isotonic, which is the 3-minute mark. So now they're starting to get balanced again. But the next one is distilled water. So this is our hypotonic solution, which means there's more water or more sodium inside of the cell. So you can see them start to break apart and disappear. That's because they are breaking or lysing within. So hopefully that makes sense. It's kind of cool. There it goes, breaking apart. Hopefully that makes a little bit more sense. Again, this is hypotonic. So I always remember hypo blows. Okay, so active transport. So the two things before I move on, the two things that we're talking about is passive transport with diffusion and osmosis. So diffusion has two parts: simple diffusion, where it just moves through it on its own, and then you have diffusion, facilitated diffusion, which needs help and usually uses those carrier proteins. Active transport, the big difference between those is that they're moving against the concentration gradient. In other words, particles are needing to move from the lower concentration back up into the high concentration. So it's kind of like me going down a slide when we're doing facilitated or osmosis, but now we went to go up the slide, and so we're going to have a lot more energy that we're including into that. So it requires energy. So anytime you see energy included into that, you're going to want to make sure that you are looking for ATP and then realizing that it is active transport because of that. So we're going from low to high, going the opposite direction. One example of this is your active transport is sodium potassium pump, where there's actually AT involved, ATP, where we are getting rid of a certain material, whether that is sodium or we're pulling in potassium, whatever the case may be. These are usually, the sodium potassium pump is actually one that is specific to sodium potassium. It actually has to fit inside of the areas for it to be able to make that exchange. And of course, ATP is included. Secondary active transport is when we're using we have two pumps. Usually there's a co-transport. So basically ions are transported out of the cell and it causes an electrochemical gradient that gives energy or builds energy up. And then the solute on the opposite side is actually sent through from a low concentration to a higher concentration. So there's our lovely little protons that are moving across that and creating that electromagnetic or electrochemical gradient. And then that creates the energy enough for something like sucrose to be able to move, or it transports the sucrose from the lower concentration to the higher concentration. So that's your secondary active transport, where one is being used to build up that electrochemical gradient, where the other one is used, and then it creates that energy to be able to pull it across on another transport area. So a co-transporter pump as well. Bulk transport, there's two pieces to that. You have large molecules and they don't fit, so you have to use a different option to get them across the membrane. And usually this is by means of vesicles. This also is required by energy. It requires energy to do it. So there's two things we're looking at: endocytosis, exocytosis. When we're looking at endocytosis, it's what's being brought in those vesicles, and then exocytosis is the materials that are being released from the cell itself. So when we're looking at endocytosis, another example would be phagocytosis. This is an example where large particles are pulled in. This is the cell membrane. It basically wraps around that large particle, and then it's pulled in with a vesicle, or a vacuole, right? And then it is pulled in whatever it needs to go within the cell, and then it's released into that area of the cell. So that's your phagocytosis, is basically eating. And then we have penocytosis, where small molecules that are dissolved in fluid are being brought in the same process. So they're basically wraps around the area that they are trying to pull in. This is the cell membrane that basically like pinches off. We have that vesicle, and then we release them into the cell. So again, this also needs energy to actually work. You have endocytosis, receptor-mediated endocytosis. So we have a receptor. So those glycoproteins on the top parts of the cell will actually send a signal out, pull it in, it grabs it, and then the vesicle still works the same way. The cell membrane wraps it, kind of takes it in, and then releases it into the area of the cell that it's needed. And again, this is where it's just binding to that receptor of the membrane. Exocytosis, it's exiting the cell. So we have substances that the cell doesn't need anymore. The cell membrane or the vesicle that's created, we talked about that last week, where it's created within the Golgi apparatus or somewhere else, where it's packaged out in vesicles and sent out to the plasma membrane. And then it's just basically released, and then that vesicle is now engulfed back into the plasma membrane, and it becomes a part of it. So that is where it's being released. Okay. Okay, so that is the lecture tonight. That was done pretty quickly. So the idea is now for you to go through the practice assignment. The one thing that gives people problems is the hypotonic. We'll go over that again next week or on Wednesday when I meet with you guys again for this lecture. And if you are having issues or you still are figuring it out, sorry. Then make sure you're writing down your questions tonight so that you can go in and ask me on Wednesday. This assignment plus everything else is open until Friday, so you have some time to revamp it if you're not sure how to do this. It's pretty straightforward. This is probably one of the easiest lectures out of all of the series that we do. So I hope you guys are doing well. I hope this helps. And again, I will see you guys on Wednesday. Enjoy your night off. And then again, if you have questions, let me know and write them down, and I will see you guys soon. Okay. Have a good night. Talk to you later.